All right, welcome to another deep dive. Today we're going to be looking at DNA replication.
Okay.
And you sent us this research paper, and it's got some really interesting stuff in here about, like, other forces that are at work besides just hydrogen bonding.
Right.
So we're going to try to unpack those together. You probably heard about hydrogen bonds holding the DNA double helix together, but this paper is all about, like, base stacking and steric effects and how sometimes they're even more important.
Yeah, it's really
Your
fascinating.
stability than hydrogen bonds.
Exactly. It's easy to get caught up in that hydrogen bonding story,
Right.
but there really is a whole symphony of forces that are kind of at play
Yeah.
during DNA replication.
Yeah.
And they often get overlooked.
So before we even get into those kind of less famous factors,
Sure.
can we just do a really quick recap of, like, DNA replication itself
Absolutely.
to get everybody on the same page?
Think of DNA replication like you're making a perfect copy of a blueprint. So you start with the original blueprint, which is your DNA double helix.
Yeah.
And you need to make an exact duplicate.
Okay.
And the star of this copying process is an enzyme called DNA polymerase.
Okay.
It carefully reads the original blueprint base by base,
Yeah.
and it adds a matching building block, or what we call a nucleotide, to create that new copy.
So it's like DNA polymerase is like the molecular architect,
It's
making sure everything's in the right place.
Yeah,
Yeah.
exactly.
It's so cool.
And what's fascinating is that DNA polymerase doesn't just randomly throw these nucleotides together.
Right.
It can actually recognize the correct base pairings,
Oh, wow.
ensuring that adenine always partners with thymine
Yeah.
and cytosine always pairs with guanine.
Yeah. I know this is kind of like fundamental stuff, but it still just blows my mind how DNA just knows how to copy itself and create all life as we know it. But I'm guessing it's
Yeah.
not as simple as just like
Yeah,
hydrogen bonds holding everything together, right?
you're right.
It can't be.
Yeah, while hydrogen bonds definitely play a role,
Okay.
your research paper really emphasizes those other forces, the base stacking and steric effects.
Okay.
Mm.
This is where I get really curious.
Okay.
Because I'll admit, I always thought that hydrogen bonds were like the stars of the show when
Sure.
it came to, like, DNA stability and accurate replication.
It's a common one.
But is that a misconception?
Yeah, we need to remember that DNA exists in a watery environment,
Okay.
and water molecules love forming hydrogen bonds too.
Oh,
So they're constantly vying for those
are they
connections
competing with the base?
with the DNA bases, yeah.
Okay.
So this means that those bonds between bases aren't as strong as we might initially think
Right.
because
Right.
the water is competing for those interactions.
So the water's almost like a meddling third wheel
You could say that.
in a DNA relationship.
Yeah, you could say that.
Wow, that's interesting.
And what's interesting is that studies have shown that even when you create DNA molecules with what we call non-polar base mimics,
Okay.
so basically bases that can't form hydrogen bonds,
Oh, wow.
they can still be replicated efficiently.
Really?
So this suggests that something else has to be contributing
Okay. Yeah, that makes sense.
to DNA stability and that accurate copying.
Yeah. So if hydrogen bonds aren't the only key player,
Right.
what about this idea of base stacking?
Okay.
Can you kind of unpack that for us?
Yeah, so imagine stacking a deck of cards.
Okay.
That's kind of what base stacking is like in DNA.
Okay.
So the bases, which are flat, aromatic molecules, are drawn to arranging themselves face to face, just like those cards.
Oh, okay.
And this stacking is driven by several forces.
Okay.
We're talking van der Waals forces, electrostatic interactions,
Wow. So it's not just one.
and even how the surrounding water molecules interact
Yeah.
with the stacked bases.
Like a bunch of different things.
It's a lot, yeah.
So it's not just like one simple force
It's not just one thing.
holding those bases together. It's like this complex combination of, like,
Yeah.
attractions and repulsions.
You got it.
That's crazy.
And here's the really fascinating part.
Okay.
Research suggests that this base stacking can sometimes be a bigger contributor
Oh, wow.
to DNA stability than hydrogen bonding.
So base stacking is kind of like the silent hero
Yeah.
of DNA
You could say that.
structure.
Yeah.
Wow, that's really interesting.
Yeah.
So what about these steric effects?
Okay.
How does, like, the size and shape of molecules
Right.
even come into play here?
So steric effects are all about how the physical shape of molecules influences how they interact.
Okay.
Think of it like trying to fit different shaped blocks into a puzzle.
Okay.
Some fit perfectly,
Yeah.
while others just don't belong.
Right.
Yeah.
So how does that, like, puzzle analogy apply to DNA replication?
So when DNA polymerase is trying to add a new nucleotide to the growing DNA strand, it needs to make sure it's the right fit.
Right.
If the wrong base tries to sneak in, you get what we call steric clashes.
Okay.
Basically, the molecules bump into each other.
Because they have the wrong shape.
Because they have the wrong shape, yeah.
Got it.
And these clashes distort the DNA helix, making it unstable.
Okay.
And more prone to errors.
So it's like trying to jam a square peg into a round hole.
Exactly.
Doesn't work.
Won't work.
Yeah.
You know, the research you sent mentions an example of this with like something called TT pairs.
Yeah.
Can you explain that?
Yeah, so thymine bases can technically pair with each other.
Okay.
But this arrangement would be really crowded within the polymerase active site.
I see.
It's like trying to squeeze two bulky suitcases into a small overhead compartment.
Yeah, there's just no space. Yeah, that limited space helps prevent that incorrect pairing from happening.
Okay, that makes sense.
Yeah.
It's amazing how these, like, tiny molecular interactions have such a huge impact on how accurately DNA gets copied.
Yeah, and it's not just the bases themselves that are involved in steric effects.
Okay.
Even water molecules play a role.
Oh, really?
Acting like tiny spacers.
Wow.
To prevent those mismatched base pairs.
So even water is part of the quality control team.
Exactly.
That's wild.
There was one study in your research that used a really large molecule called pyrene.
Okay.
Pyrene is way too big to fit opposite a natural DNA base.
Okay.
But it actually can be incorporated into DNA at places where a base is missing.
Wow.
Wow.
So the size of the molecule really, like, determines what it can even pair with.
Yeah, it just goes to show
how
the size of a molecule
polymerase active site
really determines what it can pair with inside that tightly controlled polymerase active site. Yeah.
Okay, so we've got hydrogen bonds, which maybe aren't as strong as we once thought. And then this base stacking, like silently holding things together, and finally steric effects acting like bouncers to keep things in check.
Yeah.
It sounds like these last two are really important.
Absolutely. Your research highlights how base stacking and steric effects play a much bigger role in DNA replication than we previously realized.
That's so interesting. It's like we're getting a whole new perspective on DNA replication.
We are.
Yeah.
And to really understand this shift, we need to dive into the research that specifically challenges our understanding of hydrogen bonding's role. And that's what we'll tackle next.
Awesome. I'm ready.
Yeah. So, welcome back. Last time, we were talking about how hydrogen bonds might not be as crucial for DNA replication as we once thought. Research you provided really digs into this idea using some really clever experiments with what are called non-hydrogen bonding base analogs.
These molecules that can slip into DNA but don't follow the usual hydrogen bonding rules. It's like they're going undercover to reveal the secrets of DNA replication.
I like that analogy.
Yeah.
One of the stars of this undercover operation is a molecule called defluorotoluene. Or DF for short.
DF,
And it's a remarkable mimic of thymine. One of the 4 natural DNA bases. DF is practically identical in size and shape to thymine. But here's the key difference. It can't form those classic hydrogen bonds.
So it's like thymine's twin, but with a crucial difference.
Exactly.
Yeah.
What happens when DF tries to join the DNA party?
Well, if hydrogen bonds were absolutely essential, you would expect DF to be completely rejected.
It just wouldn't.
Unable to participate in DNA replication at all.
But surprisingly, that's not what happens.
Oh, really?
Researchers found that DNA polymerase could actually insert DF opposite adenine. Just as efficiently as it would insert regular thymine.
So polymerase is like reading a language we thought was essential.
Right.
But it turns out there's a whole other level of communication going on.
Exactly.
That's so cool.
And what's even more remarkable is that DNA polymerase didn't just randomly throw DF into the mix. It specifically paired it with adenine, rejecting other bases like cytosine, guanine, and even thymine itself.
So even without those hydrogen bonds, the polymerase can still recognize, like, the correct pairing.
That's right.
It's like there's some other signal guiding the process.
There is.
Yeah.
And this finding was further supported by another experiment where researchers created a completely non-hydrogen bonding base pair using DF and another analog called DZ, which mimics adenine.
Gotcha.
This DF-DZ pair was replicated with remarkable efficiency.
Wow.
By various DNA polymerases.
So it's not just a fluke with DF.
It's kind of fluke.
Multiple polymerases can handle these non-hydrogen bonding base pairs.
Yeah, it really challenges that textbook view.
Yeah, it really does.
Of DNA replication.
Yeah.
Yeah. These findings suggest that the ability to form hydrogen bonds isn't the defining factor for DNA polymerase to do its job.
Right.
Something else must be guiding the process.
Okay, this is all, like, very thought-provoking.
Yeah.
So if hydrogen bonds aren't essential for, like, efficiency, what about their role in accuracy?
That's a great question.
Yeah.
And one that researchers are still actively exploring.
Right.
While it's clear that hydrogen bonds aren't absolutely necessary for replication to occur, they likely do play a role in fine-tuning the accuracy of the process.
So maybe hydrogen bonds are, like, part of the quality control team?
Yeah.
But they're not the only players?
They're not the only players, exactly. The research really points to base stacking and steric effects as the heavy hitters when it comes to both efficiency and fidelity. Hydrogen bonds might provide an extra layer of precision, but they're not the defining feature.
Okay, I'm starting to see DNA replication in a whole new light now.
Good.
It's not just about those textbook hydrogen bonds.
Right.
It's this, like, intricate dance of different forces with base stacking and steric effects playing, like, a much bigger role than we even imagined.
And this new understanding has some exciting implications. If we can replicate DNA without relying solely on hydrogen bonds, it opens up the question of whether we could expand the genetic code beyond the 4 natural bases.
That's a fascinating idea.
Yeah.
What would the implications of that even be?
Well, think about the potential for biotechnology and synthetic biology.
Yeah.
If we could engineer DNA with entirely new building blocks, it could revolutionize fields like medicine.
Wow.
And material science.
Yeah.
Imagine creating custom-designed organisms for specific tasks.
Wow.
Or developing new materials with unique properties based on expanded DNA.
That's crazy.
It's a whole new frontier.
Yeah.
It's really exciting.
It's amazing to think how much we're still learning.
I know.
About something as fundamental as DNA.
It is. We've explored these individual forces at play in DNA replication.
Yeah.
But now we need to consider how they all work together.
Right.
And that's where we'll head next.
Okay, welcome back to the deep dive.
Yeah.
You know, we've been talking about how hydrogen bonds aren't the only force at play in DNA replication. We've explored base stacking.
Yeah.
We've explored steric effects. But now, like, I'm really curious to understand how it all works together.
Yeah. It's like we've been looking at the individual instruments in an orchestra.
Okay.
And now it's time to kind of hear them play as a whole. Yeah. So remember how we talked about the polymerase active site being this tightly controlled environment?
Yes, like a microscopic workshop where everything has its place.
Exactly. And within that workshop, the polymerase enzyme actually creates a very specific binding pocket for the incoming nucleotide. Okay. The one that's about to be added to the growing DNA strand.
Right.
This pocket is shaped by both the enzyme itself and the template base on the original DNA strand that's being copied.
So it's like a custom-made mold that only the correct nucleotide can fit into.
That's a great way to think about it.
Okay.
And that's where those steric effects we discussed become really important.
Right.
The shape of the binding pocket, along with the shape of the incoming nucleotide, will determine if the pairing is even possible.
Right. And if it's not a perfect match, then you get those steric clashes, right? Which create instability.
Yeah, like trying to force a square block into a round hole.
It just won't work.
It just doesn't work.
And, you know, remember how we mentioned that, like, mismatched base pairs can sometimes form hydrogen bonds too?
Yeah.
But they often create, like, distortions in the DNA helix.
Well, within the polymerase active site, there simply isn't enough room
because it's so tightly controlled
for those distortions to happen.
Okay.
The enzyme is holding the DNA strands in a very precise orientation, making that steric fit even more important.
So the polymerase is almost like a chaperone.
Yeah.
Making sure everything lines up perfectly.
Yeah, that's a good way to put it.
Does this also impact, like, base stacking?
It does. This tightly controlled environment actually highlights the crucial role of base stacking as well.
Okay.
Within the polymerase active site,
Yeah.
those stacking interactions between bases become even more important for stabilizing that newly formed base pair.
It's like those stack cards we talked about earlier, but now someone's holding the stack steady to make sure it doesn't topple over.
Yeah. The polymerase enzyme enhances those stacking interactions, making them a key contributor to the overall accuracy and efficiency of DNA replication.
So it truly is a beautifully orchestrated interplay of forces.
It is.
It's not just about hydrogen bonds anymore. We need to think about base stacking, steric effects, and how they all work together.
It all connect
within the polymerase active site.
Yeah, and what's truly amazing is that this entire process is happening constantly within ourselves with incredible speed and accuracy.
It's wild.
It really highlights the elegance and efficiency of nature's design.
It makes you wonder if we could ever replicate this level of sophistication in the lab.
It's a huge challenge.
Yeah.
But one that scientists are actively pursuing. The more we understand these fundamental forces, the closer we get to designing and engineering new molecules that can interact with DNA in very specific and predictable ways.
That's amazing.
Yeah, it's really cool.
This deep dive has completely changed how I think about DNA replication.
I'm glad to hear that.
It's not just about those textbook hydrogen bonds anymore. It's this incredible dance of forces that makes life possible.
It just goes to show that even in something as seemingly simple as DNA copying itself, there's a universe of complexity and wonder waiting to be explored.
Thank you so much for guiding us through this deep dive.
You're welcome.
I feel like I've gained a whole new appreciation for the elegance and complexity of DNA replication.
Me too.
And for our listeners, we hope this has sparked your curiosity about the hidden forces that shape the world around us.
Yeah, keep exploring.
Until next time,